Okay, this chapter is over nutrition, metabolism, and body temperature regulation. So a nutrient is a substance that the body uses for growth, maintenance, and repair. And there are three major groups of nutrients, and these make up the bulk of ingested food. So you have your carbohydrates, you have your lipids, and you have your proteins. And there are other nutrients that you need in small amounts. So that would include vitamins and minerals, and because water is a requirement, technically it's also a nutrient. So this slide talks about appetite enhancers and appetite suppressors. I am not going to hold you responsible for these. You can spend time learning about what all these are and how they work. But the hunger control center, I want you to keep in mind, is in the hypothalamus. That's where we have these neurons that can enhance appetite or suppress it. So the one that I want you to really pay attention to and remember is ghrelin, because ghrelin is a very important appetite enhancer and is produced by the stomach. It makes you feel hungry. So that's the one. Pay attention to ghrelin, appetite stimulation. Okay, so we're going to talk about different nutritional food groups, starting with carbohydrates. But before we do, I want to talk about the word calorie. Now, the calorie is the amount of heat energy that will raise the temperature of 1 gram of water 1 degree Celsius. Now, that doesn't mean much to us, but I just want you to know that when you're breaking down foods that you eat and you're breaking them down into their individual building blocks, you're releasing a lot of heat energy in the process, and this heat energy is measured as calories. So carbohydrates have about 4 calories per gram. Now, if you consider that the average person consumes about 2,000 calories per day, and this is just average, it varies from person to person, dietary requirements, the recommended dietary allowance is about 125 to 175 grams per day. Now, this is about minimum 500 calories. And you do need a minimum amount, 100 grams a day, to be able to maintain adequate glucose levels, and you need a minimum of 120 grams per day to fuel the brain and nervous tissue. So the brain requires glucose as a source of fuel. That's very important to remember. It's also important to remember that glucose is the body's readily available source of fuel. The recommended intake should be 45 to 65% of total caloric intake, and you want this to mostly be complex carbohydrates in the form of whole grains and vegetables. Now, carbohydrate makes up about a pound in the average human body and is stored mostly in 3 places. But most of it, I want you to remember, 325 grams is in the muscle as glycogen. That's the storage molecule for glucose. So most of your carbohydrates stored in muscle. 90 to 110 grams in the liver as glycogen. So the second place is as glycogen in the liver, and then the rest of it is found as blood glucose. And just remember that nervous tissue, red blood cells depend nearly exclusively on carbohydrates as their source of fuel. Now, excess carbohydrate can be converted into fat, so you got to be careful with excess carbohydrate because it can seriously affect weight. Glucose is the molecule ultimately used by the body cells to make ATP. So you want to make sure that you get enough carbohydrates, especially if you are active. Now, a century ago, this is on the second page, about a third of the way down. A century ago, Americans consumed an average of 4 pounds of sugar per year. Now Americans ingest on average 200 to 300 grams of carb per day. That's 3/4 of a pound a day. And note that a single non-diet soft drink contains 38 to 43 grams, or about 8 teaspoons of sugar per 12-ounce serving. That's a lot of sugar. Complex carbohydrates, starches are found in bread, cereal, flour, pasta, nuts, and potatoes. Simple carbs are called sugars, are found in soft drinks, candy, fruit, and ice cream. Now, an important type of carbohydrate that we don't digest is fiber, primarily cellulose that's found in plant cell walls, but we use this. It absorbs water and it increases the bulk of feces. So it helps to soften the stool, and it can increase its bulk by as much as 100%. This increase in bulk will stretch the colon and the reflexes to contract, so it increases peristaltic movement, which quickens the passage of feces from the colon. But too much fiber can interfere with the absorption of certain nutrients, so there's a balance. Going on to lipids. Lipids are the most dense form of nutrients, 9 calories per gram. And dietary sources, the main fat is called the triglyceride, which is your neutral fat. And keep this one in mind because it does break into glycerol and fatty acids, and that's going to be important when we talk about diabetes later on. So there are essential fatty acids, linoleic and linolenic acid, but most people get enough fat, they don't have to worry about that. But dietary fats are important because they help absorb your fat-soluble vitamins A, D, E, and K, and they also are an energy source for your liver cells, your hepatocytes, and also for skeletal muscle. They are the component of the myelin sheath, and they make up cell membranes, because remember, your cell membranes are made up of phospholipids. So, the American Heart Association has said that fats should make up less than 30% of total caloric intake, saturated fats 10% or less, and your cholesterol should be no more than 30 milligrams per day. Now, the goal is to keep blood levels of cholesterol lower than 200 milligrams per deciliter. And the typical American diet, greater than 40% of the calories taken in come from fat. And they add up fast because 9 calories per gram, and if the average American consumes 30 to 150 grams of fat a day, 150 grams of fat a day equates to 1,350 calories. And they obtain 50% of their calories from fat, and I'm willing to bet that most Americans, it's probably 60% or more. So cholesterol is another type of fat that is the structural basis of bile salts. Your steroid hormones come from cholesterol, remember, and vitamin D is a cholesterol-derived product. So it's the major component of plasma membranes. 15% of cholesterol is ingested, okay? So keep that in mind. Only 15% comes from the diet. The rest is made in the liver. So if you have a problem with too much cholesterol, usually they will prescribe medications or statins that will stop the liver from producing cholesterol, but there are some serious side effects involved with a lot of those medications. Cholesterol is lost from the body when it's catabolized or secreted into bile salts. Now, this is how fats are transported. They're transported by what are called lipoproteins. Now, your VLDLs are very low-density lipoproteins. These are molecules that have mostly triglycerides, and they transport triglycerides from the liver to the peripheral tissue cells, mostly adipose tissue. And you want to remember these by definition, what they do. LDLs, low-density lipoproteins, they transport cholesterol to the peripheral tissues for membrane storage and hormone synthesis. And your HDLs, they're the good guys because they leave the liver empty and they bring cholesterol and fat back to the liver to be metabolized, so they help to prevent plaque buildup inside your blood vessel walls. And they also provide cholesterol to steroid-producing organs that require cholesterol for making those hormones. So here's a picture of a breakdown of what these particular substances have, what their makeup is. And there's one added here. This is a chylomicron. Now, we talked about this back in Chapter 23 in the digestive tract chapter when I was talking about lacteals. And I said that lacteals transport fat. When fat's broken down in the small intestine and then it goes into the lining of the small intestine, those cells in the lining package up that fat with a little coating of protein and absorb it into the lacteals, and then the lacteals take it through the lymphatic circulation, and then it goes to the adipose tissue. But if you see here, mostly triglyceride, not much else. So those are your least dense form of lipoprotein, and that's how fat's transported through the lacteals. Here's your VLDLs, mostly triglyceride, then your LDLs, cholesterols, the main component, and then over here, mostly protein and a little bit of cholesterol and some fat. So you just want to remember generally what these three are involved in doing. This one is fat to peripheral tissue cells. This one is cholesterol to peripheral tissue cells. This one leaves the liver empty, brings the cholesterol mainly back to the liver to be broken down. So we said earlier, total cholesterol, this is blood levels of cholesterol, should be 200 milligrams per deciliter or less. And anything greater than 200 milligrams per deciliter is linked to plaque buildup, or what we call atherosclerosis. Now, the form in which the cholesterol is transported in the blood is important to measure, and sometimes these things change. These values are constantly being looked at by the health associations and to see what they consider a good or poor value. But right now, anything greater than 60 for HDLs is considered good. Anything less than 40, not so good. High LDLs are associated with cholesterol deposits in vessels, buildup of plaque, which is atherosclerosis. 100 or less is good. Greater than 130, not so good. So now we move on to proteins. Now proteins, like carbohydrates, are 4 calories per gram. And the recommended daily allowance is about 44 to 60 grams per day. So you know what that is? There's 30 grams to an ounce. So that's about 2 ounces of protein. So I don't know of too many people that eat a steak that's only 2 ounces, but that's all you need per day. And that's about the size of a deck of cards. So enjoy your little deck of cards worth of steak when you go out and you're actually getting your RDA. But if you are me, it's going to be a lot bigger than that. So we get enough protein. So your dietary sources, you have 2 types, complete and incomplete proteins. So complete proteins come from eggs, milk, fish, most meats, soybeans. They contain all the essential amino acids. Now, your vegetable sources are incomplete because they're lacking some of these essential amino acids. So legumes, nuts, cereals, a lot of your vegetables do not have the essential amino acids. So you have to make the right combination. If you're a vegetarian, you probably already know that you have to put certain types of vegetable proteins together to get the complete complement. So here's a picture showing you the breakdown of the amino acids and the ones that are essential, and then showing you corn and other grains. These are the ones that they have. So they have tryptophan and methionine, and beans and other legumes, they don't have those, but they have isoleucine and lysine that corn and other grains don't have. So if you complement them, you'll get the complete assortment of the essential amino acids. So when you're using amino acids have nitrogen, and carbohydrates and fats don't have that. So they talk about this nitrogen balance. And you want to make sure that in the body, the rate of protein synthesis equals the rate of breakdown and loss. And if you have a positive nitrogen balance, that means that the synthesis exceeds the breakdown. And this is normal in children, pregnant women, tissue repair. Negative nitrogen balance is where the breakdown exceeds synthesis, and that happens when you have a lot of stress, burns, infection, injury, poor dietary problems, starvation. It's just a term you might hear used, but I won't test you on anything about nitrogen balance. So what do you use amino acids for? You use them for producing hormones. A lot of your hormones are protein-based. And so anabolic hormones, growth hormones, sex hormones accelerate protein growth and synthesis of proteins. So adrenal glucocorticoids released during stress, they cause proteins to be broken down and converting those proteins, those amino acids, into glucose. So this is how you use amino acids. Okay, so now we're going to go to minerals. So there are 7 minerals that are required in moderate amounts. So you have calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium. These are the major ones, and there's others required in trace amounts, and they work with nutrients to ensure proper body functioning. And a lot of these are used as cofactors and enzymatic functions, but this is beyond the scope of what we need to know and involves a lot of biochemistry, so we're not really going to talk about that. But uptake and excretion is balanced to prevent toxic overload. Now, vitamins, there's 2 types based on solubility. Make sure you know the water-soluble vitamins, the B complex and C, are absorbed with water. B12 absorption requires intrinsic factor that's produced by the stomach, and these are not stored in the body. Anything that's not used within an hour is excreted, so megadoses are absolutely useless. Fat-soluble vitamins A, D, E, and K. These are absorbed with lipid digestion products, so you have to have lipids to absorb these, and they're stored in the body with the exception of vitamin K. Now, there are pathologies due to excess because these can be stored in tissues. They're fat-soluble. Now, we're going to be talking about something that involves the breakdown of your proteins, carbohydrates, and fats. This can get pretty complex. I'm going to try my best to make it understandable for you, but we're going to walk through this slowly and make sure that we can understand this as we're going through it. But your proteins, carbohydrates, and fats, first of all, let's look at what proteins are made up of, amino acids. So digestion in the GI tract lumen to absorbable forms, and they're going to be transporting these breakdown products via the blood to the tissue cells. And the breakdown products of proteins are amino acids. So that's what you're going to be transporting. Carbohydrates, glucose, and other sugars. Fats, the major fat is a triglyceride, so remember the breakdown product glycerol and fatty acids. So this is what happens when you digest the food. And then the cells take those breakdown products, and the cells can make them into proteins. Amino acids can go into proteins. You can have in the liver and then skeletal muscle, you can take those glucose molecules and make them into glucose and store them. And remember, when you're putting things together, that's an anabolic reaction. When you're breaking them apart, it's catabolic. You can take the fats. The adipose tissue can take the fats, the glycerol, and the fatty acids and make fats. So we know that can happen. That's what the cells can do. But the cells can also take molecules, especially glucose, and go through a process called glycolysis, break that glucose into 2 pieces, and those 2 pieces are called pyruvic acid, and then use the pyruvic acid and take it into the mitochondria and make ATP. And ATP is what the cells really need to drive their metabolic processes. This is the gasoline that drives the cells, okay? So you have to have ATP. So oxidative breakdown occurs in the mitochondria. So these 2 processes occur in the mitochondria. Remember that. And your products of this process, by breaking down the glucose, your products are going to be carbon dioxide, water, and a lot of ATP. So, breaking down carbohydrates, proteins, and fats in the digestive tract. Those breakdown products can be taken into cells and restructured and stored as proteins, glycogen, and fats, but they can go through this process and make ATP because you need ATP to be able to drive the machinery that does all this. Okay, so this process is what we're really going to spend some time talking about. So this is called the oxidation of glucose. Oxidation means to break it down. So here's your glucose molecule right here, and it requires oxygen. And the products are water, carbohydrate, and look at that, 36 ATPs and a lot of heat. Remember I said when you're breaking down food products or food molecules, you're releasing heat. So these are calories, okay? So you're burning glucose and you're burning a lot of calories. So that's important. And a lot of ATP. So there are 3 processes involved with glucose metabolism. Glycolysis, that word means splitting the glucose. Krebs cycle, and electron transport chain and oxidative phosphorylation. Please stay with me, and if you have questions, I will be glad to address those. This takes place in the cytoplasm of the cell. These 2 take place in the mitochondria, which is the powerhouse, remember? So this picture here shows you basically glycolysis. So what happens is you're taking that glucose molecule, and I don't know if you can see this here, but there's 6 little balls. That means it has 6 carbons. And when you break it apart, and I'm not going to go over all the details here because I'm just going to make this simple for you, you wind up with 2 pieces. And these 2 pieces have 3 carbons. So you're going from 1 6-carbon molecule to 2 pieces that have 3 each. This is called glycolysis, splitting the glucose in half. And what you wind up with here is by taking the glucose and splitting it in half, you have 2 pyruvic acid molecules, and you're going to make 2 ATPs, and you're going to have 2 of these molecules that are called nicotinamine adenine dinucleotide. That's a mouthful. But NADH, okay, is really important because that is made of something called niacin. Now, niacin is vitamin B3. So on the notes I have you, or on the review sheet, I have one line where I say, make sure you know the vitamins that go into the oxidation of glucose. And I'm going to show you where some of these vitamins are used. So vitamin B3, and a lot of these are B-complex. So you wonder what B-complex vitamins do? Well, I'll show you where some of them are. So NADH has niacin, vitamin B3, in its structure. So you produce 2 of those, and you produce 2 of these. Now these are high energy intermediates, because when you break glucose in half, you're splitting it in half, you're releasing a lot of energy. And some of these H's come off, and they go right here. They get picked up by this molecule, and they get transported, okay? So these are like taxi cabs. They're taking passengers from this molecule that's broken apart, and they're going to carry them away. So the end product of this glycolysis reaction is 2 pyruvic acids. Now, if you have no oxygen present, this is anaerobic, that goes into lactic acid. So think about this. When you're exercising and you're full steam ahead, going as hard as you can, you're limited to the amount of time you're going to be able to perform that exercise, because without available oxygen, you're going to build up lactic acid. Lactic acid drops the pH of the muscles, makes the muscles burn, and eventually you stop, usually within a very quick period of time because of the lactic acid buildup. Now, with oxygen, you're not going to build up the lactic acid because the pyruvic acid is going to be taken into the mitochondria and used in the Krebs cycle and electron transport chain to make a lot of ATP. Without the oxygen, that pyruvic acid goes into lactic acid. So very important concept to know. With oxygen, aerobic, if you are exercising moderately, running slow, jogging, you can go a long time because you've got a lot of oxygen there, and you can transport this pyruvic acid into the mitochondria and make a lot of ATP, and you can keep going for a long, long time, okay? So glycolysis, the final products of glycolysis, splitting the glucose, 2 pyruvic acid, don't worry about the formula. You get 2 of those nicotinamide adenine dinucleotide, okay? They picked up some passengers there, okay? So they got some H's on there from the glucose, and you make 2 ATPs. Now, for glycolysis to continue, the NADH must be present to accept hydrogen atoms. So those empty taxicabs have to be there to pick up the passengers, the H's from the glucose molecule. And we're going to see where they come in later on, because those taxicabs are going to be dropping off their passengers at the electron transport chain. So stay tuned. Okay, now this is the Krebs cycle. And remember, this happens in the mitochondria. So that 3-carbon pyruvic acid that's produced from glycolysis, it requires oxygen to move any further. So here's what happens. Oxygen is going to bind with one of those carbons on the pyruvic acid, okay? So the molecule that does this is a derivative of thiamine, which is vitamin B1. So vitamin B1 is needed to take that carbon off the pyruvic acid, combine it with oxygen, and you get the formation of a CO2, okay? Now you just have 2 carbons left. That's called an acetyl group. And that acetyl group is going to bind to this little transport molecule, the blue thing that's called coenzyme A. Now, coenzyme A has a vitamin in its structure too, and it's called pantothenic acid, which is B5. So look at this. We've already had B3 niacin, that's an NAD. We had B1 thiamine, that's the decarboxylation molecule involved with taking a carbon off of pyruvic acid and adding it to oxygen, okay? And now we just had B5, that's pantothenic acid. That's in coenzyme A. So you've got all these B vitamins floating around. You wonder what they did? They help to make ATP. Isn't this fascinating? And by breaking the carbon off, the hydrogens that are associated with the molecule, another taxicab. Okay, so another NADH comes along, picks up some H's, and is going to carry them off. And remember, we're going to talk about where the taxicab passengers are dropped off when we get to the electron transport chain. So you see there's a whole bunch of crap going on here, okay? I'm going to make this simple. All right, so here's your acetyl-CoA. 2 carbons attached to coenzyme A, which is like a little escort. And the escort's going to take the acetyl group and it's going to attach it to the pickup molecule of the Krebs cycle. There's a pickup man there that's going to pick up this acetyl group from the enzyme. And that guy is called oxaloacetic acid. And he has 4 carbons. So he says, come on, acetyl group, join to me. And the coenzyme then can get, because enzymes, remember, are not consumed in reactions, they make the reaction go faster. So the escort, the coenzyme A, drops off the 2-carbon acetyl group and goes back and picks up another one. So he's just recycled. He keeps getting more and more guests and bringing them in. So the pickup molecule that has 4 carbons combined with the 2-carbon acetyl group, that becomes citric acid. And citric acid is really the first product of this reaction, and sometimes instead of the Krebs cycle you will see this cycle called the citric acid cycle. So it goes around and round and round and round and round and round and round, and you can see in the process, here's your products. You got another taxicab company here picking up more H's, you got another taxicab company here, you got another one here, and then you got this other taxicab. This is a totally different company, and it's called flavin adenine dinucleotide. Flavin. What B vitamin do you think goes into that one? Aha, riboflavin. Riboflavin is vitamin B2, which goes into that particular molecule. So, 3 of these, NADH plus H, and then 1 FADH2 from the competitive taxi company, flavin adenine dinucleotide. Okay, and then you also have 1 ATP, and look at this. The 2 carbons, okay, the 2 carbons, they come off right here. They get combined with oxygen, and they form CO2, so you have 2 more CO2s there. Now, I want you to keep in mind that after glycolysis, remember, we have 2 pyruvic acids from glycolysis, right? So you really need 2 turns of the Krebs cycle to get both pyruvic acids through this process. So by the time you get the 2 pyruvic acids through from 1 glucose molecule, all of this doubles. So you wind up with 1, 2, 3, 6 NADHs, 2 FADH2s, and 4 carbon dioxides and 2 ATPs. Because you got to double it. You got 2 pieces to put through the process. And remember, this takes place in the mitochondria. It requires oxygen. You have to have oxygen to be able to bind to these carbons. All right, this is the electron transport chain. And the electron transport chain is where the taxi cabs come. They drop off their passengers. They drop off their H's, and the H's get pumped through these proteins, and they build up a proton gradient. Because remember, they're all positive charged. Okay, so you're dropping off all the passengers, and you're pushing them into the terminal, and they don't like to be close together. Okay, so you're packing these like charges together, and like charges want to repel each other. So you're building up a lot of pressure. And the only way that they can get through is that they have to come through this little channel. That's the only way that they can get through. And that channel is called an ATP synthase. So when the hydrogens start to build up pressure and they rush through this molecule has a channel going through the middle, and it has like a rotor that spins when the hydrogens rush through. And that's called chemiosmosis. So it uses the protons. A hydrogen ion is called a proton. And when these things rush through, it generates the energy necessary to spin the rotor, open the binding sites for ATP, ADP, and phosphate. Those two together make ATP. So this enzyme is important for generating ATP, and it does it because of the current by the flow of hydrogen ions that rush through the channel and open up the binding sites for ADP and phosphate. So that's really, really important. And so I want to just mention one thing. Oxidative phosphorylation. What does that mean? We talked about oxidation of glucose. You're oxidizing glucose, breaking it down. And by breaking down glucose, you are going to phosphorylate an ADP. Okay, so that's why they call it oxidative phosphorylation. Oxidizing glucose, phosphorylating ATP, ADP, and creating ATP. Very, very important. So the complete oxidation of glucose, here's the whole thing, the whole picture. Glycolysis. Okay, glucose gets split into 2-pyruvic acid. And you get the generation of 2 NADH and 2 ATPs. And then you get the conversion of pyruvate, which is also another name for pyruvic acid, into acetyl-CoA. And by doing that, you have to strip off a carbon that gets combined with oxygen, and then you produce 2 NADHs there because you have 2 pyruvic acid, so 1 for each pyruvic acid. Then your 2 acetyl-CoAs go through the Krebs cycle. So each one goes through, that's 2 turns, 6 NADHs, 2 FADH2, 4 carbon dioxide, and then the passage of those H's, you got 10 NADH total, 2 FADH2. Each NADH is going to produce 3 ATPs, and then FADH2 produces 2, and we'll talk about that in a minute. But you have 10 NADHs, 2 FADH2s, 6 CO2s, and from the Krebs cycle we got another ATP, so there's 2 there. And then by the electron transport chain we wind up with 6 water molecules and 34 ATPs, which gives you a total of 38. So here's where the ATPs come in. NADH2 gives you 3 ATPs. FADH2 gives you 2. And so that's where the total, how you get the number, okay? So if you look at the notes, it walks you through the entire process on page 4, on page 5. And it talks you through the whole process. go to page 6 of the notes, and it says you get 38 total, okay? Because overview of ATP production, you have 10 NADHs, so each one gives you 3, so that's 30 total. FADH2, each one gives you 2, so that's a total of 4. Glycolysis, you generated 2 ATPs, and the Krebs cycle, you generated 2, 1 from each turn, which gives you a total of 38. But since the NADH is produced during glycolysis, they're out in the cytoplasm. They have to cross over into the mitochondria. They expend some energy, so you expend 2 ATPs to cross those NADHs to get them into the mitochondria. So you just have to subtract 2, and that gives you a total of 36 ATPs, which is theoretically the number commonly used to complete the glucose oxidation equation. So that's where your 36 comes from. Now we're going to talk about glycogen metabolism. So the average adult body, remember, has about 400 to 450 grams of glycogen. Most of it's in the liver, 1/4 of it's in the liver, 3/4 over there in the skeletal muscle, and small amounts are in cardiac muscle. But glycogen is formed. So the word glycogenesis, you want to make sure you just know what the definition is. Now, I did put the enzymes here, but you do not have to know the enzymes, but I just want you to be aware that most of the processes that occur in the body require an enzyme. So when you are digesting food and you're taking food in by way of the digestive tract, the first thing that the cells do is they're going to fill up their ATP stores. So you go through glycolysis, Krebs cycle, electron transport chain, and the cells are going to make ATP and fill up those stores. And then after that, then whatever's left over goes into glycogen. So that's glycogenesis. Mainly occurs in the liver and skeletal muscle. Glycogenolysis is the breakdown of glycogen. Now that's in response to low blood levels of glucose. So in between meals, you're going to break down glycogen, convert it into glucose, and keep blood glucose levels normal. Gluconeogenesis, that word means creating glucose from non-carbohydrate sources. So if you don't have enough glucose, what you're going to start to use is you're going to start to use proteins and lipids to make glucose. So glucose is formed in the liver from glycerol, okay, that's part of the fat molecule, and amino acids when blood glucose levels drop. So it protects the body against damaging effects of hypoglycemia. And remember, because nervous tissue requires glucose, it's really important to make sure that you have enough glucose to drive the nervous system. So the liver, we talked about the liver in chapter 23, but there's a lot of functions. And actually these hepatocytes, they have over 500 metabolic functions. Synthesizing bile. Bile is our fat emulsifier to help us absorb fat. Packaging up fatty acids to be transported. So remember we talked about those lipid proteins. Synthesizing plasma proteins. So a lot of your proteins are made in the liver. Forming non-essential amino acids. So there's 12 of them that can be made in the liver. 8 are essential and have to be taken in by way of the diet. Proteins, when they're broken down, are actually going to be converted into that nitrogen group in the protein will be converted into ammonia and then to urea. And we'll talk about that in a minute. The liver stores glycogen, and it also stores vitamins, iron, and is a major detoxification center in the body. Because remember, nutrients from the GI tract, from the stomach and from the intestines, they go through the hepatic portal vein to the liver. So all your medications, alcohol, food, everything, toxins go to the liver first. So the liver gets a hit. It gets all those toxins and it has to be able to detoxify those things. Okay, so lipid metabolism. There are two words, lipogenesis and lipolysis. Now, when you have when you have digested food, okay, and you've broken it down into your triglycerides are broken into glycerol and fatty acids, that's lipolysis, breakdown of fat. Lipogenesis is where you're putting fats together. So building fat would be anabolic, catabolic, breaking it down. So here is fat stored in adipose tissue, and there's your dietary fats. Those are your main sources of fat. Those are triglycerides primarily. So breaking it down, lipolysis, building it up, lipogenesis, okay? So I want you to follow along with this because this really helps understand where ketogenesis comes from and ketoacidosis that happens in diabetics. So lipolysis, you're breaking the fat into glycerol and fatty acids. Glycerol has 3 carbons. And if you put 3 carbon molecules together, you can make a glucose, right? So by taking that glycerol here into glycolysis, you can actually take that part of the molecule and you can make glucose if you have to. Remember, gluconeogenesis. How is that defined? Getting carbohydrate from non-carbohydrate sources. So fats can be converted. The small part of the fat, the glycerol molecule, can be converted into glucose. But here's the problem. The main part of the fat molecule are these long fatty acid chains, and they can be in excess of 20 carbons apiece, and you have 3 of them. Now, these fatty acid chains are broke down 2 carbons at a time by a process called beta oxidation. So you chop them up into 2 carbons. Now remember, your 2 carbons come in here in the form of an acetyl group, CoA. And see how this arrow only goes one way? You can't use those long fatty acid chains to make glucose. They only make the acetyl group. And where does that go? Into the Krebs cycle, electron transport chain to make ATP. So, when you break down the fat molecule, you're going to generate a lot of these. You could have as many as 60 or more of these, one glycerol, and you need 2 of those to make 1 glucose. So you might be talking about 120 acetyl groups, and there's no way to do anything with them except to put it here. When your ATP stores are filled, you know what happens? Those acetyl groups go into ketone body formation. So here's what happens. When a diabetic doesn't have insulin to take the glucose into the cells, they rely on lipids. And there's only a small part of this molecule that can make glucose. But because there's so many of these that get broken down to so many of these, now you start to produce ketone bodies, and ketone bodies, well, they're acids, and they can put a person into metabolic acidosis. There you go. That's how it happens. Now, a few of these can go into making some cholesterol, which will produce steroids and bile salts. But boy, I will tell you, there is your problem with too many fats. So accumulation of ketones in the blood results in ketosis. Ketones are acidic, and they result in metabolic acidosis. And when people are in ketosis, the breath smells fruity. Ketones have a fruity smell to them, and that's the vaporizing of the acetone. And the compensation is to breathe rapidly to release CO2, because CO2 levels increase or decrease pH. So by getting rid of CO2, you'll raise the pH. And ketosis is common in starvation, unwise dieting, people that don't eat the right amount of nutrients, and also diabetes. Ketone bodies are excreted in urine, so you can pick it up there in a urinalysis test. So protein metabolism. Protein metabolism, excess dietary protein results in amino acids being used for energy. And just to simplify this for you, what happens is, this is the group that you can't do much with. Your proteins have this amine group on it. It's an NH2 group. So by a process called deamination, this is on the review sheet, you're going to be taking this group off and converting it to ammonia. And ammonia then is added with carbon dioxide to make urea, and urea goes out in urine. And a person that has a failing liver, because all this happens in the liver, you can smell the ammonia because you can't complete this process. The liver's not functioning. So in a failing liver, you will see this level build up, and it really does smell. So that's what happens to proteins. Taking off that amine group and then converting that to ammonia, and then it goes to urea, out is urine. That's the normal process. So this is the interconversion of carbohydrates, fats, and proteins, just to sum everything up. Glycogen converted to glucose, and then you have these different intermediates that are formed and eventually split. Glycolysis splits it into pyruvic acid. Take off a carbon of that 3-carbon molecule, you got an acetyl group, combine it with CoA, goes into the Krebs cycle. Okay, that's how carbohydrates do, how they are brought through this process. Proteins. Proteins are broken into amino acids, okay? And then by taking off the amine group and converting it to ammonia, what's left is called a keto acid. Now, those intermediates can go right in here. They can go into the glycolysis process, and some of them, depending on their structure, because you have 20 different amino acids, some of them go in here, okay, depending on what they are. So proteins can be used to make ATP by going here or here. And remember, ammonia goes to urea. Fats, broken into glycerol and fatty acids. Now remember, the glycerol molecule can only go here, okay? It goes here. Fatty acids, only here, because this arrow does not go backwards. So acetyl group into the Krebs cycle. Too many ketone bodies. And pyruvic acid, if you don't have oxygen, you can't go here. You go into lactic acid production. That's anaerobic. This is aerobic. I hope by going through this a couple of different ways, this is kind of getting a little bit easier to understand. Now, there are 2 states I want to talk about. This happens when you're digesting a meal, okay, and you're absorbing the end products. Anabolism exceeds catabolism. You're absorbing nutrients, the cells are taking in the amino acids, the simple sugars, the glucose, and the breakdown products of fat, and you're storing these substances. So carbohydrates, remember, glucose is the major cellular energy for fuel. Glucose is converted in the liver to glycogen, and it can be converted into fat. Synthesized fat and protein released to the blood for storage by adipose tissues as very low density lipoproteins. Remember, we talked about those. They transport fat to be stored by adipose tissue cells. So what I want you to remember is this. In the absorptive stage, the first thing that happens in the cells is that ATP stores are filled. Okay, so you're going to store ATP. The next thing that happens is you're going to store glycogen. Okay, and then whatever's left over, you're going to store as fat. So that's what really happens to carbohydrate. ATP, glycogen, fat. You make proteins. You take the amino acids and you make proteins, and glycerol and fatty acids are formed into triglycerides. So this is the absorptive states, anabolic processes. And the hormone that drives this, insulin. Insulin drives the uptake of glucose and also the uptake of amino acids into cells. And what happens in the liver? Amino acids that are in excess can be deaminated. They can be used for energy or stored as fat, and they can be converted into keto acids by taking off that amine group, and they can go into triglyceride formation, or they can be used to make ATP. That's the absorptive stage. Post-absorptive state, catabolism rules here, because what's happening here is you're breaking down the products that you've stored. So this is in between meals, okay, in between meals. And the hormone that's important here is glucagon. Glucagon is the hormone that drives this process. So now you're going to break down the fat, break down the glycogen, break down the proteins, and catabolism exceeds anabolism. So you're not making fat, glycogen, and protein anymore. Now you're using the products that you stored during the absorptive state and you're breaking them down. So the goal is to maintain blood glucose levels, keep it steady in between meals. So these processes make glucose available to the blood. So lipolysis, glycogenolysis, and if necessary, gluconeogenesis. If you don't have enough carbohydrate, then you start breaking down your proteins and your fats, okay, to make glucose if you need it, to make ATP. So promotes the use of fats for energy, glucose sparing, and to save glucose for organs that need it. So remember, nervous tissue requires it. So that's what you're going to do. So, major metabolic thrust, ATP, is created by glucose. And if you don't have enough glucose, you don't have enough glycogen, then gluconeogenesis, you can use fatty acids from that triglyceride molecule, and ketones that you get, ketones are produced from excess fatty acids. They can be produced if you make too many, but your major metabolic thrust is catabolism, breaking proteins down into amino acids, glycogen into glucose, triglycerides into glycerol and fatty acids, and the liver, if necessary, can take those amino acids, convert them to keto acids, and use them as glucose to drive processes to make ATP. There. All right, so now let's go to basal metabolic rate. Metabolic rate reflects the energy the body needs to perform its most essential activities. It's usually measured in the post-absorptive state, meaning nothing orally for 12 hours, so fasting for 12 hours, in a reclining position, relaxed mentally and physically, room temperature 20 to 25 degrees Celsius, which is normal room temperature, and recorded as kilocalories, that's our nutritional calorie, per square meter of body surface, kilocalories per square meter per hour. And the average 70 kilogram or 150 pound adult basal metabolic rate is about 66 kilocalories per hour, and that's how many calories you need just to survive. And it comes in at around 1,560, somewhere around there, calories per day. And on the notes on page 9, about halfway down, it talks about the average number we're using, 2,000 calories a day. So just to survive, 1,500 calories a day. But this basal metabolic rate is influenced by things like body surface area, your age, your gender, body temperature, stress, thyroxine production, which consumes more calories. Thyroxine increases metabolic rate. So all these things are something that needs to be considered when you're looking at this number. So factors that influence BMR: it increases as the ratio of body surface area to volume increases. It decreases with age. Males have more muscle mass, so they have a disproportionately higher BMR. Increases with temperature or stress, and thyroxine increases oxygen consumption, cellular respiration, and BMR. So body temperature regulation, normal body temperature is 37 plus or minus 5 degrees. That equates to roughly 98.6. Enzymes function optimally at this temperature. When you increase the temperature too high, it denatures the proteins and interferes with the function of neurons. Children less than 5 years of age, convulsions can occur at 106 degrees Fahrenheit. The limit for life is 109 degrees. I've never seen it. Tissues tolerate low body temperatures extremely well, however. So the core is where most of your heat is located. So that's like in the central part of the body, the axial part. The skull, the thoracic, abdominal cavities has the highest temperature. So rectal temperature is the best clinical indicator. And the core temperature is regulated kept fairly constant. And the blood's the major agent of heat exchange. You radiate heat off from the surface of the body. And the skin, the shell, has the lowest temperature, and it can fluctuate between 20 and 40 degrees Celsius. So these are heat production, heat loss mechanisms. So heat production, basal metabolism. Remember, when you're burning glucose, heat's a byproduct. When you're cold, shivering moves muscles, and by burning glucose, making those small movements, that's going to generate some heat. Thyroxine, epinephrine, they have stimulating effects on metabolic rate and temperature effect. Warmer cells are going to metabolize faster, and they're going to produce more heat. Heat loss. You radiate heat away from the surface of the skin. Conduction, convection, and evaporation. And by evaporating liquids off the surface of the skin, by sweating, it carries away a lot of heat. So the homeostatic imbalance, hyperthermia, if the temperature gets too high, it can depress the hypothalamus, which is the thermostat, which controls temperature. And if you depress the thermostat, then that creates a positive feedback mechanism, which can make the temperature continue and to increase, and that begins at the core, and the increased temperature makes the skin hot and dry, and eventually, because increased temperature denatures proteins, and the organs can be damaged, and sometimes it can be irreversible. Heat exhaustion, not quite so bad, but it's heat-associated collapse after vigorous exercise, not getting enough water, and by losing water volume, your blood pressure can drop. Heat loss mechanisms are still functional, however, but it may progress to heat stroke if the person is not cooled and rehydrated properly. Fever is controlled hyperthermia due to infection, cancer, allergies, central nervous system injuries can cause it. Remember that these pyrogens, we talked about these in the immune chapter, they reset the thermostat to increase the temperature, and prostaglandin E is what causes the increase in